In this video, we'll be talking about the use of biotechnology in agriculture, and especially it has come as a great help in developing pest resistant crops. Now, here we are going to use the technology in manipulating the DNA. We all know that DNA codes, or during the process of transcription, it forms mRNA. And this mRNA is actually a code to synthesize a particular kind of a protein. This is a specific sequence, and this specific sequence leads to the production of the protein through the ribosomes. This is the normal mechanism, and it is this protein that actually helps in various kinds of activities of the organism. Now here we are using or manipulating this DNA to silence or to have the stopping of the synthesis of a particular protein. Now how this is brought about? Now there are several actually when we are talking of the pest resistant crops, this technology has come as a savior, and it can be called as genetic pesticide. Now here there are nematodes which are actually causing several kinds of diseases in plants and animals, and also in the human beings. Now we'll be talking about a specific kind of nematode, that is Meladogyne incognitia. It is found in the roots of tobacco plants. So it infects tobacco plants, and as a result it causes lot of damage. That means the yield is highly affected. Now here the biotechnology comes as a rescue. Here the infestation or the infection and the damage was controlled by the method of RNAi. So this infection by this particular nematode in the tobacco plant can be checked by RNAi, and this RNAi is RNA interference. Let us talk about and try and understand what is RNAi. RNAi actually is a biological process. It is found in all organism, and in this RNAi actually it inhibits gene expression by neutralizing specific mRNA molecules. So how RNAi functions is, since we know that mRNA is using for ribosome for the synthesis of the protein, this RNAi affects the mRNA, and when it affects the mRNA, it will stop the synthesis of that particular protein, because this is again a specific gene sequence. So this RNAi will bind to this mRNA, will modify it, and the protein synthesis can be stopped. Now this method, it is a very common this is a very commonly found RNA interference strand, which is found as a control mechanism or defense mechanism. So it helps in silencing the gene. So RNAi actually regulates by gene silencing. And since this is happening after the transcription, it is known as post-transcriptional gene silencing. So at this is it is after the transcription, so we call it as post-transcriptional gene silencing. Or in short, we call it as PTGS. Now, this has been used to manipulate or to prevent the infection due to the nematode. Now, how this method is utilized, we are going to study over here. Now, the RNA, as I told you, is found in all eukaryotes to defend, and it is a double-stranded DNA. Now, this nematode, the scientists have actually isolated the gene that produces proteins for the growth of the nematodes. Now, for the survival of the nematode, this protein is very, very essential. So, the nematode has a specific gene that codes for protein, and this protein is responsible for causing disease. This has been identified. Now, what scientists have done, the scientists have isolated the gene from agrobacterium vectors. Now, here we are using agrobacterium vectors. Agrobacterium vectors are nematode specific. That means they will be acting on specific, or they will be effective against specific nematode only. And these are why we are using these vectors, because they are small size plasmids. One example is Agrobacterium tumefaciens. That is Agrobacterium this has come as an effective one. Now, what it has done is, this agrobacterium is in this isolated gene from here, it is introduced, this is as a vector. So, this is now introduced into the nematode. Sorry, it is introduced into the plant. This is introduced into the plant. Now, in the plant, the transcription and the translation will take place to form double-stranded DNA. Now, it produces the sense and antisense RNA. So, when it is introduced into that is when we are talking of agrobacterium vectors, we are actually introducing the double-stranded DNA over here. This double-stranded DNA codes for sense and antisense RNA. Now, these two RNA are complementary to each other, and they form double-stranded RNA. So, it is now double-stranded RNA. This double-stranded RNA, that is RNAi, it binds with the gene that causes the synthesis of this particular protein. Now, since it has it binds with the specific area, or this gene which is coding for the particular protein which caused the disease, it actually is interfering or stopping the synthesis of this particular protein. As a result, the protein will not be produced, and since this protein will not be produced, the nematode will not be able to survive. So here there is a specific interfering RNA that acts on this particular nematode and does not allow it to survive. Now, why does this dsRNA binds to mRNA? Because when we are talking of special gene, that is the mRNA. Why this double-stranded RNA binds with the mRNA? It is because the dsRNA is complementary to mRNA and it prevents the translation of mRNA. So this dsRNA is actually complementary to the mRNA, and since it will be binding to it, it will obviously affect the coding, and that code will that code which is responsible for producing the disease causing protein will be affected, and therefore it is silencing the mRNA. So when it binds over here, this causes silencing of the gene. This is silencing of gene, and as a result the protein is not synthesized. The complementary RNA, this complementary RNA can be from an infection causing virus with RNA genomes or mobile genetic elements, that is transporons, and they replicate through the RNA intermediate. Now this has come as a great advantage because here we are using RNAi to develop or a defense mechanism against the specific nematode, and therefore we are calling it as a genetic pesticide. RNAi is therefore a genetic pesticide. So this has come as a great help because here the use of chemicals are reduced and we are manipulating the gene to develop the resistance against certain pests. Now this is a very common use in the case of when we are talking of the nematode infestation in the case of tobacco plant, and therefore the plant gets protected itself from the effect of the parasite.